Hi, everyone. Thank you so much for being here today. We're very fortunate to have Dr. Stewart Campbell from Axial Therapeutics with us today. Thank you so much for joining us.
Pleasure to be here. I'm happy to have the opportunity. So thank you for the invitation.
I'm just going to give a little bit of a background for the audience about your company, how you guys kind of came to be, and then feel free just to tell us everything, get as scientific as you like, and we're excited to hear from you. So.
Terrific.
Dr. Stewart Campbell is from Axial Therapeutics. It's a clinical stage biopharmaceutical company that's dedicated to improving the lives of people with neurological diseases. Axial is advancing a pipeline of small molecule drugs with defined mechanisms of action that act on new targets to mitigate the impact of metabolites and bacteria in the gut linked to neurological diseases, pathology, progression, and symptoms. The company's drug discovery approach is rooted in the groundbreaking research of its co-founder and chief scientist, Dr. Sarkis Mazmanian at the California Institute of Technology, who has seen clear biological links implicating the gut microbiome via the gut-brain axis in serious neurological conditions. Axial's lead candidate, AB2004, is a gut-restricted molecular therapy for irritability in children with autism, which of course is where we're very interested to hear about. So thank you again, and please feel free to start.
Great, so I'm ready to go. So if you call the slides up, I can get started.
Okay.
Great, so thanks again, Jessica, for that nice introduction to the company, and thanks to the organizers for the opportunity to tell you a little bit about why we're so excited about what we're doing in autism. And we're a private company, but we have to be thoughtful about what we say and how we say things, and so this is mandated by the powers that be. So we'll skip that and get to the good stuff. So as you mentioned, Jessica, what we're really trying to do is change the way we think about and treat neurological conditions, and in particular neurodevelopmental disorders, and we have some work going on in neurodegenerative disease as well. And some early stage work in oncology, but it's really trying to target these indications by understanding how the bacteria and microbes that live in our gut can interact with the human host, and in particular how the brain and the gut microbes communicate. And so there's a lot of press out there on the gut microbiome and how one should try to manage it and how it can be good for you or bad for you. We at our core are serious scientists. We take a very rigid, rigorous scientific approach to everything we do. And that comes directly from our scientific founder, Sarkis Mazmanian at Caltech, who's really a world-renowned expert in the gut microbiome, and in particular, the gut-brain axis, or that mechanism of communication between the gut and the brain. And you'll hear a lot of other avenues that people are exploring to try to take advantage of the relationship between the gut and the brain. And most of those to date focus on things like fecal matter transplant, or adding single microbes to try to shift the microbiome in a particular way. And those have shown some remarkable results, and we are highly supportive of those efforts. Our approach is just different in that we try to take all that information that we can garner from what's going on with the gut microbes and what's going on with things like behavior or brain function, and we try to distill it down into very specific mechanisms of action. So for us as a drug discovery team with a lot of experience in discovery and development of drugs, we really need to understand anything we bring to the clinic. We need to understand how it's acting. We need to understand that mechanism, which means we need to understand the target. So the output of our approach here is new defined drug targets, molecular targets in the gut. They could be on microbes, they could be on the human tissue, but something in the gut against which we develop small molecule therapeutics that are designed to act in the gut and actually just go through the gut without being systemically absorbed. And we think there's some inherent advantages on several levels that this can provide. And it's really a completely different approach to what I think anybody has seen before. And I hope to show you, through our example with AB 2004 in autism, exactly what we mean and why we're so excited. So maybe just a short primer on what we mean by the so-called microbiome gut-brain axis. So if we think about how the gut and the brain can communicate with one another, we have all the gut microbiota in our GI tract, and that's trillions of bacteria, hundreds of different bacterial species. In fact, we have as many gut bacteria living in us as we do total human cells in our body. So, one could argue we're 50% bacteria and 50% human. And we've evolved to have a very symbiotic relationship with the gut microbiota that live within us. Sometimes things can get out of whack, and we can end up with problems that result from that. And that's where we focus. And so we think about the different avenues that the body uses to have the gut microbes and the brain communicate. And let me see if I can grab a pointer here. So we have sort of what we call the hardwired relationship between the gut and the brain, and that's through the enteric nervous system. So the enteric nervous system is that set of nerves and the whole system that is surrounding all the tissue in our gut, and it controls things like telling us when we're hungry, when we're full, are we digesting food properly, is there an invader present? And this can all be done directly through a hardwired communication between the gut and the brain. We're actually leveraging this in our Parkinson's program because there's a very clear link between certain bacteria in the gut and Parkinson's disease that we believe is operating through the enteric nervous system. For the case in point today, the autism work, we're really focused on a different avenue of communication between the gut and the brain, and that's through the peripheral circulation. And what we mean by that is we have gut microbiota, as I said, trillions of bacteria. They're very busy. They help digest food and fiber and so on. And they're constantly cranking out small molecule metabolites. And in fact, if you took a blood sample today, and you measured the metabolites in there, roughly half of those metabolites that are circulating at any time actually have a microbial origin. So we would be dead without the microbes. The challenge here is what happens when we get too much of a set of metabolites or too little? How does that translate into some kind of change in our health? And so for autism, and I hope to show you exactly what we mean in a few slides, but what we mean is that the metabolites actually that are produced in the gut can get across the gut barrier into the blood, and they can travel to the brain through the blood. And there they can exert all kinds of positive and negative effects. And this is where we focus on in our autism program. So for this group, we probably don't need to give you a background on autism, so we'll kind of give you a pass on that one for this talk. But as I mentioned, the autism program is really focused on this avenue of communication between the gut and the brain. And so, part of our motivation here is that there really are no current treatments for core behaviors, and the only two approved drugs with an autism label approved in the US are two atypical antipsychotics for the treatment of irritability associated with autism. And we'll get into this a little bit, but these antipsychotics actually have some significant side effects that actually, in our opinion, have limited their use in the field. So there's clearly an opportunity here and an unmet need that we think we can fill. And so our motivation here and our rationale for going down this road was, again, no approved drugs in the areas that we're focused in. And it started really with a realization that GI disturbances are highly prevalent in the autism community. And then that led to some work, not necessarily by us, but several groups started to look at the gut microbiome in children with autism. And consistently have found that the gut microbiome profile of kids with autism is different from their matched control peers. The challenge here is it's not always the same type of different. And we all know this sort of N of one, and every child with autism is unique. So too, you could say, is their microbiome. And so this presents some challenges here, but if you just look at the identity of the microbes that are present, you may not get to all the clues you need in order to make a drug discovery program out of it. So we started focusing on not just who the microbes are that are present, but what they actually do. And what this does is it eliminates, or at least reduces, the confounding factor of functional redundancy in the microbiome. So you could have two completely different microbiome profiles and have essentially the same functions being executed. So we focus on what the microbiota actually make, and that's these bacterial-derived metabolites. And what we've shown, and several others as well, is that these metabolites are actually altered in children with autism. And we've published a couple of papers in that regard. And that was really a big clue for us that maybe there's something going on with these metabolites. And so our whole mechanistic hypothesis here is that there is a class of what we call neuroactive microbial metabolites that contribute somehow to autism-like behavior phenotypes. And we believe this is happening by these metabolites having an effect on myelin plasticity in the brain. And I'll comment a little bit more specifically on that later. So the idea here is if this is actually what's underlying some of these behaviors, how do we actually develop an intervention? So our treatment hypothesis is really to just reduce these neuroactive microbial metabolites in the gut so that we can see systemic reductions in those metabolites, which would then in turn lead to lower brain exposure, which in turn would lead to improvements in behavior. And so we've done a lot of work, some of which I'll summarize in a moment, in mice, showing that our molecule AB2004 is actually able to bind these metabolites in the gut and reduces their levels in circulation, and that translates into improved behavior. And so the first clue here was work coming out of Sarkis Mazmanian's lab at Caltech, published already 8 years ago. So we've been at this for a while. And what he noticed in some mouse work that he had been doing with the late Paul Patterson at Caltech was in a mouse model in which the mice, when they're pregnant, they're given a mock infection to sort of induce an inflammation response. And if you do it at the right time, the pups that come out of those mice have a much higher probability of exhibiting autism-like symptoms. Deficits in communication, deficits in anxiety, in social behaviors, and so on. And what they noticed in these mice, these ones that had been, where their immune system had been activated, and those offspring actually had significantly altered metabolite levels. So you take the blood out of the mice, you send them out to a company that will profile hundreds or thousands of metabolites, and you literally look at the differences between control groups and the affected groups. And what we found was that the most dysregulated metabolite was this small molecule called 4-EPS. It's 4-ethylphenyl sulfate. And I'll show you a little bit more about that as well. What we showed is that the mice that had high levels of 4-ethylphenyl sulfate, that correlated with these autism-like behaviors in these mice. So what the next experiment was to take synthetic 4-ethylphenyl sulfate and literally just inject it into wild type or normal mice and see what happened. And what they showed was that daily injections of one shot of 4-EPS every day for 3 weeks led to a profound anxiety phenotype. And there's just one example here of some of the data here. And what we're showing here is that the mice exposed to 4-ethylphenyl sulfate, when you put them in an arena, an open field, it's essentially a box with no cover, and you monitor by video how frequently and how much time the mice will venture from the safety of the corner or the wall out into the open in the middle. And this is a surrogate measure for anxiety. So mice that are anxious tend to stay against the wall or in the corner for fear of being out in the open. So when you see a reduction in the number of times or the amount of time they spend in the center, that's an indication that the mice are exhibiting some anxious behavior. But we thought injecting 4-EPS into a mouse is not how the mouse would normally be exposed to 4-EPS. So what we next did, we fast forward, sorry, I'll come back to this one. We fast forward a few years, and we start to focus on several different metabolites. And we said, there's got to be a way to give the mice more natural exposure through the gut in a way that would mimic how the microbiome would do it. So we said, well, why not just create a microbiome that we could engineer and then stick into the mice so that the mice would be chronically exposed in a more natural setting to these metabolites. And so we look at molecules here like 4-ethylphenol, paracresol, and indoxil, shown here in the middle. These are the metabolites that the gut microbes will produce by breaking down protein into the component amino acids like tryptophan and tyrosine. And you can see that this component here of the molecule looks the same as this one. So tryptophan gets converted by the microbes into this indoxil. Tyrosine, you see this motif here in the molecule, looks similar to this one and this one. So these two will come from tyrosine. And what happens is we all have these metabolites, by the way. It's just that in the case of autism, certain subsets of people with autism will have higher levels of these metabolites. And what we know is that these molecules, because of their physical properties, are very rapidly absorbed into the liver. And the liver identifies them as foreign substances and tries to get rid of them. And what it does, one mechanism it uses to do that, is to put in this case, a sulfate group on the molecule. So you see these sulfated molecules. Now 4-ethylphenol becomes 4-ethylphenyl sulfate, paracresol becomes paracresol sulfate, and so on. And these are the molecules that we see in circulation. These are the molecules we see getting into the brains of mice. And these are the molecules that we believe are impacting changes in myelination in the brain and causing behavioral changes. And how do we know that? So when we take normal mice and we install microbes that are very efficient at converting tyrosine into 4-ethylphenol, we see really high levels of the corresponding 4-EPS in the circulation and the brains of those mice. And what we see is that when we compare the mice that have chronic exposure to 4-EPS in this case, to the ones that the same microbes are present, but they've just been engineered so they cannot produce this molecule. So you have your positive and negative controls. And we see that the ones that are exposed show decreased vocalization. We measure this by ultrasonic communication between the mice. And again, reproduce this anxiety phenotype. So they spend less time in the open areas, in this case of the elevated plus maze. So we have an anxious phenotype with minimal communication. There are several other aspects as well that we show are produced by things like 4-EPS. So we said, okay, here's mechanistic information that these molecules that come from the microbes are actually actively contributing to the behavioral differences between those exposed and not exposed. So this is our first clue that there's some reason to go after these metabolites and try to reduce them. The concept being, if we can reduce them, can we actually turn a group that looks like this into a group that looks like this? So what we did, apologize for the density of this, but it's important to walk through this. So what we did is we took those same mice and we, again, showed that the mice that have the right microbial composition in their gut produce whopping levels of 4-EPS. We see this in their blood and in the urine as well. And those that don't have the right microbes, we don't see any 4-EPS. So here's our negative and positive control. So then we took this molecule we now call AB2004, and we said, rather than try to modify the microbes with a drug, let's just bind up the metabolites in the gut. So you take this drug, you take it orally, it passes through the GI tract, and you pass it in your stool. But as it transits through the GI, it picks up molecules like 4-EPS and paracresol and indoxyl sulfate, and it sequesters them. So then they're excreted in the stool. So then that should lead to reduced levels in the blood and urine. And we see this in these mice. And now we have a system that actually allows us to test the effect of the drug and test the idea of reducing these metabolites in systemic circulation. And what we show here is that the mice is the same 4 groups of mice. So we have those mice that are on a control diet and those that are on a diet that is doped with AB2004. And we see here that the mice that this is kind of our benchmark, so-called normal control mice. And we see in the case of the mice that are exposed to 4-EPS, we see a significant anxiety phenotype. So they spend a lot less time in the center of the field. And then if we compare the second bar to the fourth bar, we see that the AB2004 is actually able to help recover the anxiety phenotype. So we see this move up. And this group is statistically significantly different from this group. So we've made a jump here. Similarly, there's several ways we can measure this type of anxiety. We have the same pattern where in this particular measurement, the mice that show significant anxiety are rescued completely by AB2004. So this is anxiety. Then we did a measure of another slightly different measure of anxiety, but is also reflective of some of the stereotype repetitive behavior. And this is the marble burying test. So you lay out these large flat marbles on the top of the bedding, and mice, being very curious, will bury the marbles, and then they'll quickly move on to some other task, unless they have this stereotyped repetitive behavior phenotype, in which case they will bury many more, statistically more marbles than their control counterparts. And what we see is that AB2004 was able to rescue the repetitive behavior phenotype. And also we see the same thing on self-grooming, which is another repetitive behavior anxiety measurement. So we've got good preclinical proof of concept, admittedly in this highly contrived reductionist system. And so, but this did give us the motivation to take AB2004 into the clinic. And so what we did was we went into an adolescent male autism population. We did this study in 2019 and 2020. So we got this done just before the pandemic hit, thankfully. And this was done in Australia and New Zealand. And it was a safety and tolerability study, really focused on whether AB2004, which remember stays in the gut, there's no systemic exposure. So we expected a strong safety profile. But we need to verify this. We're going into kids who are vulnerable, who, we have to be very cautious here. So we did a very careful dose titration. So we started them all on a low dose, and after 2 weeks, they graduated to a medium, and then after another 2 weeks, graduated to a high dose, all based on safety measurements here. So they didn't progress if they didn't pass the safety tests. I'm happy to say that everybody, except for one child who had some other issues, made it past into the high dose group. So we had no significant, no drug-related adverse events in this study. So very, very safe profile so far, and knock wood that we continue to see that as we move ahead in the clinic. So while it was a safety and tolerability study, we also measured the things where we hope our drug will move, which are these bacterial metabolites. So we measured them in plasma and in urine. And we had a whole library of about 10 of them, not just the one or two that we've been talking about so far. But we also took the opportunity to measure a battery of behaviors. So irritability, anxiety, adaptive behavior with the Vineland, repetitive behavior, et cetera. And the reason we did this is one of the challenges with things like autism and other neurodevelopmental disorders is it's not a given that what you see in mice will translate directly into the human population. So something that might show up as anxiety in mice may show up somehow differently in the human setting. So we cast a very wide net and we measured several behaviors to make sure that if there was any kind of efficacy signal, that we would see it. And so the results from this study far exceeded our expectations. And so one example we show here, remember in the mice, we had a very strong anxiety phenotype that was induced by exposure to a microbial metabolite, in that case 4-EPS. And when we reduced it with the drug, we rescued that anxiety phenotype. So when we look at the whole cohort of individuals, and we measure anxiety using the Pediatric Anxiety Rating Scale, or PARS, we saw at the full cohort level, we saw a trend, but it wasn't significant. But when we looked at the group that started out with an anxiety score above 10 on this scale, and we didn't arbitrarily set 10. That's imposed by how that anxiety scale was developed. And so this is recognized as kind of a floor for autism. So if you're below 10, you really don't, in autism, they can't really detect whether you really don't have anxiety. So, now when we looked at those who had what we would call high anxiety going into the study, we saw a really significant reduction in the anxiety scores. And what we don't show it in this graph, but we took another measurement of 4 weeks after they stopped the 8 weeks of treatment, and the improvement persisted. We don't know if it goes past that, but as the last time point we took, the effect on anxiety held. What's interesting is out of all the metabolites that we measured, we saw significant reductions in almost all of them. We saw, some going down 60, 70% reduction. And that's in the whole group. But then when we look at this subgroup, the high anxiety subgroup, we see reductions again, but the pattern is different. And so if anyone is a data scientist or is into machine learning or AI, this presents a really nice problem to dig into, where if you could understand the relationship between these patterns of change and these changes in behavior, we may be able to come up with things that are diagnostic or are indicative of a change, or at least allow us, maybe inform the next round of research into how metabolites could impact behavior. So we're quite excited about that. So anxiety is great, and it's an important problem in autism. But one of the strongest signals we saw was in irritability. And this is measured by the Aberrant Behavior Checklist, which is a very standard, well-established instrument to measure aberrant behaviors. The irritability subdomain was used actually as the instrument to measure change in irritability for the drugs aripiprazole and risperidone, which are those two atypical antipsychotics that are approved to treat irritability in autism. So when we looked at the full cohort and we saw significant reductions, this is the same set of bars that we showed on the previous slide. But when we looked at the full cohort, even though we did not include people based on their irritability score, we saw a significant reduction in irritability across the board. And when we did a subgroup analysis post hoc, and we show that for those who entered the study with an anxiety score above 15, which is considered the irritability clinical threshold. So these kids are pretty anxious, right? And you get up into the 30s and 40s, and those kids are probably on the borderline for being admitted to a hospital for treatment. And what we see with this group is an even more pronounced improvement in anxiety. So those with high anxiety, or sorry, irritability. So those with high irritability coming in, we actually were able to get their average scores down below the severity threshold. We were thrilled to see this. I'll make a couple of extra points here. Number one, this was an open label study. So if any of you are familiar with how clinical trials are run, an open label study means there's no placebo control group. And we are fully aware of the potential for a placebo effect when you're measuring behavior. The nice part about this particular effect on irritability is that the instrument used, the aberrant behavior checklist, is so well characterized that we know roughly what the size of the placebo effect is. And it's somewhere in the 3 to 6 point range. So it cannot account for the effect we're seeing. So we believe firmly that we have a strong treatment effect, certainly worth looking at in a future clinical trial. So our goal here now, when we saw this irritability score or change, we said, we have an opportunity here to actually move this drug quickly through the clinic, and with a known regulatory pathway. And I'm sure you're all aware of some of the challenges that other groups have had trying to get drugs approved for autism and failing at the pivotal point. So it's clearly a complex path. So anything we can do to de-risk the development of this drug, and we should do. And the irritability presents a known path, both from a clinical perspective and from a regulatory perspective. FDA is very familiar with irritability and how to measure it, even in the autism population. So we said, let's go after irritability because it is the least risky path to get the drug approved. And then we can expand the label from there. We can look at anxiety, we can look at communication and these other things. But this gives us an opportunity to get the drug quickly to approval and get it into the autism community because it has such a strong safety profile. So we're very excited about where this is going. So one of the, another note here on why we think the drug is actually working the way we think it is. I mentioned that we did another measurement 4 weeks after the drug was stopped. In the case of irritability, what we see is that 4 weeks after we remove the drug, this effect on irritability starts to reverse. It's still lower than the baseline, but it's started to rebound. And what we see at the metabolite level, this was the same graph we showed before, is that this is at the end of the treatment period. So at the end of the 8 weeks, we measure their metabolites. We take the drug away and we see all the metabolites pretty much go back to baseline. So we essentially reverse the effect on metabolites and we start to see a reversal of the behavior that goes along with it. So this toggling of metabolites tracks with toggling of behavior. So we're very confident that we're actually seeing something real and new here. So where do we go from here? Irritability is a huge indication. It's a huge unmet need. More than half of the kids with autism will be presented to a doctor for treatment of irritability at some point in their life, typically in that puberty type of time frame when the kids become bigger and stronger and their emotions are going up and down. They can become challenging to handle. And so that's typically when parents will take their child in for some kind of intervention. Unfortunately, interventions available are not great, and we hope to change that. So where we're going next is this phase 2b efficacy study. So this trial will be placebo-controlled. We'll have a full placebo group, and we'll look at 2 different dose levels of AB 2004. And in this case, we will include kids, so it's going to be a fairly big trial, 195, spread evenly across the 3 groups. There will be male and female teenagers with autism, and we'll confirm the diagnosis of autism of everybody coming in the study. They will have to have an irritability score of 18 or higher, so fairly high up on the irritability scale. and obviously have to have some GI symptoms as well, given the microbiome relationship. So it will be 8 weeks on drug, 4 weeks off drug, and we'll measure irritability at several points along the way. And we'll measure several other things: anxiety, metabolite levels, obviously safety, changes in the gut microbiome over time, changes in GI function over time. So we hope to get a much more comprehensive view of how changes in metabolites can, how do they track with changes in behavior, how quickly can they come down, how low can we get them, and just try to understand the drug much better, which we hope will set us up for pivotal phase 3 studies in the near future. So this trial, you can find it on clinicaltrials.gov now. Just search under AB-2004 and it should come up. And we hope to have this trial read out in the first quarter of 2023, so about 18 months from now. So that was a speed talk through our story in autism, and just to thank everybody who was involved with all this preclinical and clinical work. And I hope that this gives you an idea of how we took some human data, went back into mice, developed a hypothesis, tested it, found a drug, and then took that back into people. And that whole process is something we hope to be able to do again and again in different programs. So thank you for the opportunity to speak, and I'd be happy to answer any questions.
That was wonderful. Thank you so much for that, for the breakdown of your study. So I have a couple of questions that popped up, and hopefully we can go through them. And if you have anything else to add, definitely let me know. So by looking through everything and all the mechanisms of action and how it works, is incredible. And it's really interesting to see the whole 4-EPS, the p-cresol, because obviously these are big problems in the ASD community, and the sulfate issue. I have a question actually regarding what was when you're testing for this, for your first trial, and now for your second trial, which I think is underway, and then obviously moving forward, is there a level of autism that you are, has a cutoff? Obviously you need to do an age bracket, but is there also a level of severity where people can...
Look, we debated that very intensely here. And I think right now the short answer is no. There is no cutoff for severity. We measure it, and we're also measuring functional level, sort of the PEDS-QL, the quality of life, the WASI, which is, I guess, some people would call it an IQ test. I hate using that term, but so you kind of get an idea of intellectual disability level. We're just capturing that information. We're taking anybody who on the spectrum, regardless of how severe or high functioning they might be. And the reason for that is we don't know yet whether the drug works better in one subset or another. And that's one of the things we want to find out.
No, it makes sense though. Yeah, I think I love that you have a broad, kind of like a broad brush right now, just to really see. And then you can also see what changes are possibly made, let's say in IQ, let's say in the severity of symptoms that you're not specifically targeting, but that are symptoms in autism.
Exactly.
So that could be really interesting. And then to kind of branch it off into groups could be, even more get even more specific on dosing and every single thing. So I think that's amazing. So obviously you've done phase 1. We're doing, well, I say we, the proverbial we.
We're all together in this.
And then you were mentioning also phase 3. And I think that phase 2 should be rounding up around 2023, if I'm not mistaken.
That's correct. Yeah.
So, and in terms of you were mentioning getting things kind of pushed out quicker. So we're not, with autism, it always seems like it gets somewhere and then it doesn't. And then we're all just kind of sitting here oh, that was fun. So what are your thoughts? One of the questions from Dolly was, when will the medication or supplement be possibly available to either be prescribed or I'm assuming prescribed, but to the public? What are your thoughts? A ballpark.
Yeah, sure. So first, clarify, we are developing this as a prescription drug, so it will be prescribed as versus a supplement. So that I just want to make sure that's clear. And because we're going that path, we have to go through all the rigorous clinical trials and collect all the right data, especially to make sure it's safe and effective. And you notice this trial that we're doing next all the trials so far have been in teenagers. We've had discussions with FDA, and we both want to get into the younger kids. Turns out you can diagnose irritability as young as 5 years old. So our goal would be in phase 3 to capture 5-year-olds up to 18-year-olds, right? And so the way it would go is we finish the phase 2 that in 2023, we go to FDA with our data and we say, this is what we want to do in phase 3. The phase 3 study is not going to be much bigger than the study we're doing now. And at least the way we have it thought through now. And so that would take another couple of years to get done. So if you project out and assuming everything reads out and it continues to be safe and we can demonstrate it's safe and effective, we're looking at somewhere in the 2026 timeframe.
That's honestly not bad.
It's not that bad. I mean, it's still, I know it's hard for people to hear that and they have to wait longer and they want answers now and they want help now. We are 24/7 here, going as fast as we can, but we also have to do it right. And it does take time.
No, absolutely. And is that, in terms of getting that, just that rounded number of 2026, is that more for like full approval, or are we talking like, is it possible? Like you've basically shown, I mean, obviously we have to go through more trials, but that it's safe. Now we have to show that it's continually effective and hopefully continually safe. So with that being said, is there at one point, like if you finish phase 2, are you allowed to like put it out for compassionate care, for example?
We right now we can't say that we will be able to do anything like that. We are constantly looking at all of those options, but this is really, anything like that would have to be discussed very openly with FDA, and we'd have to have complete buy-in from them before we would consider doing anything like that. The best answer I think I could give is we'll get the data and we'll look at how compelling the data is in this phase 2 study, and we'll revisit that question, but it's not up to us. It's really up to the FDA if we want to go down that road.
Absolutely. I always like to ask the tough questions because the parents are, as I am, I think since I've been born, I want it now.
Yeah.
So there's that. Just really quick, Crystal did have a question because you were mentioning kind of to go backwards and work on the kids 5 and older, which obviously is very important, something that's always pushed with ASD treatment is that, the younger you can kind of help the kids, the better. But also, is there a cutoff that you had mentioned up to 18? So is that your cutoff is 18?
Yeah, because I think for now it is. There's a lot we don't know. And there's a lot of dogma out there that, like you said, you need to get to them early and there's less you can do as they get older. That's probably true for some things, but we know that the brain is a very plastic organ, way more plastic than we knew or really understood even 5 years ago. And so I think that there is the potential for this to even be used in adults, but we wanted to make sure that we captured the pediatric population, because if we can get to them early and help them, maybe this will help them be focused in school more, or there'll be less behavioral issues in school. There's all these sort of potential cascade consequences that could come from going earlier, but we still think this drug may have some merit to be used in adults. We would just have to do all the trials to prove that. But yeah.
No, 100%. I think it's amazing. I mean, just seeing everything that's broken down. I know Enrique wants to jump on and ask a question as well.
Yeah, no, thank you, Stu, for that presentation. That was fantastic. I just want to say a couple of things. Like the first one is that, like, just so people understand the magnitude of what you're talking about, like your research could give us like two things at once, right? So on one hand, we're getting a potential treatment for ASD. At the same time, we're actually getting a biomarker, which at this point we don't really have for autism. So, I mean, like proving that we would have like these biomarkers, a single biomarker. There are obviously like secondary biomarkers. We have multiple of those, but we don't actually have a single one, and this could be it. So the significance of that is really tremendous. But you also mentioned something that is like super interesting. You actually kind of mentioned that like a little quickly, and I just kind of want to give people an understanding of the magnitude of what you were saying is that this is kind of giving us probably a glimpse into something that could be much bigger, right?
Yes.
So like by identifying these metabolites, the promise of that from the data science perspective, from the machine learning perspective, from the artificial intelligence perspective, is just something that at this point I don't want to kind of step into your research, but we're really just kind of scratching the surface into this, quite frankly. So AB 2004 is, I'm guessing from your perspective, guys, this is just kind of the stepping stone, right, into this path of research that we could be kind of collective and we're kind of looking forward to kind of collaborating with you guys in any way, shape, or form we can. But I think that's just tremendous. So I guess my question is, I suppose you guys are using like this is just the beginning, so to speak, yes. Right? On your research.
It is, and well said, and I appreciate those comments. I think once we started to learn about these metabolites, I think it took a while for it to really sink in the magnitude of what we were seeing in terms of the potential and both in our understanding. And one of the things actually I didn't get to, because I was so sensitive to the time, was we've actually taken those mouse experiments we talked about. We actually took the brain tissue out of those mice, and we showed that 4-EPS, that metabolite, actually arrests the development of a type of cell called an oligodendrocyte. And oligodendrocytes are the cells that make myelin. And you think of myelin as kind of the insulation on a wire, right? So you have an electrical wire and you have the plastic cover on it. That insulates the wire from being short-circuited. And so myelin does that for neurons. The cells that make myelin are oligodendrocytes. 4-EPS, when it gets to the brain, arrests oligodendrocyte development. Doesn't kill the cells, it just stops their development. So when you remove the 4-EPS, these oligodendrocytes go through their normal development again, and you can reestablish the potential to make myelin. And if you think about why that's just such a fundamental process in the brain for everybody, not just people with autism, but every time you learn, you go to a piano lesson, you study something in school, you need to make new connections in your brain or strengthen existing connections. That requires myelin. And if you have a myelination deficit, those connections are going to get either messed up or not made in the appropriate way, and that can translate into behavioral changes. So I didn't get to that, but you're absolutely right. The more we can understand about these metabolites and their impact in the brain, the more we can learn not just about certain behaviors within autism, but we can go to a wide variety of neurodevelopmental disorders.
Yeah, and also there's this research that I'm sure you're aware of, because we have actually had Dr. Jorgen Hahn doing a presentation with us, not so long ago, and he also kind of used this route. It was not specifically for treatment development; it was more like for a diagnostics type of tool. And it was like a similar type of data science exercise where like there were like a small set of metabolites that would, that could possibly explain. And I guess what I'm going with my comment is not only like the potential for developing treatments, for identifying biomarkers, but also the potential for preventing, the development of autism. So, I mean, to the extent that we're able to really narrow down what is the ultimate consequence in terms of like these, the pathogenesis that may be, GI related or something else, but to the extent we can kind of sequester or block these processes, we would also be in a position to hopefully be able to prevent more cases in the future, is it not?
I think that's a possibility. I mean, it's obviously there's a lot we would need to do research-wise there, but we can see, what steps we would need to take to begin to figure that out. So, as you said, this is really the tip of the iceberg for us, and maybe we're seeing what's above the water, and the 90+% of it is below the surface that we haven't scratched through yet.
But it's amazing what you're showing, especially like when you're speaking about irritability and anxiety. Most of the irritability, I was like, oh, I could use this. But just showing like that your numbers, you can't, you can absolutely see. Sometimes you see, sometimes it's so small what the improvement is, what's at least been able to show on data. And so it was really nice to see that it was a pretty dramatic drop in anxiety and irritability, which, like you said, I mean, when it comes to other neurological disorders, I mean, I also like that you touched on the fact that, of course, everyone always pushes for the younger kids because it is a big deal, of course,
Yep.
in anything, help kids when they're little. But, thankfully we know how plastic the brain is. How they used to think after 13 or something, you can't do anything.
Exactly. Yeah.
We know that, that's completely been false. It's false, it's fake news. You can totally, people have made amazing advancements in as an adult in their 60s, even they can improve.
Sure.
So I think that's an amazing thing that you guys are, like you said, on the cusp, there's something there and we're really excited about it. There was a question from Dolly. I'm going to try to ask this. It's interesting. She said, I think she's talking about the percentage of difference between mice and humans. She said, which is the percentage of similar results when you do experiments on mice when you repeat in humans? Which percent of comparable success between species? And obviously mice and humans are not the same. So there is, there's a safety thing you do and then you move forward too.
Yeah.
But I think the question is more, maybe what did you see like in the percentage of mice and the percentage of humans, like for the improvement very
Yeah. So, wow, that's I commend the question because I've never had that question that way before, but it's a very difficult question to answer. So I guarantee you whatever I say is not going to satisfy the person who asked the question, but I'll do my best. So I think, when you're measuring behavior and trying to in an animal model, and then you try to translate that to human, one has to have pretty low expectations that will translate as the same kind of behavior in a human, right? And I'll give you an example. So in Rett syndrome, for example, which used to be part of the autism DSM-5 classification, it's not anymore, but there's a hand issue with kids with Rett syndrome. And one of the mouse models will often have a clasping phenotype. And people have argued, oh, maybe that's indicative of what the hand gestures might be like in Rett syndrome. It's absolutely not the case. So I think we have to be careful about making those things. So that's just kind of a general comment. Specifically, if the question was, 80% of the mice responded to 2004, or 80% of the mice showed behavioral alteration when exposed to the metabolite, and what percentage was that in the human? What I can tell you with irritability, we had about almost a 90% response. So just about everybody had a favorable change. We had one kid who did not in that first study. And so we had a very high response rate. Whether that holds in a bigger placebo-controlled study, we're about to find out. And so that seemed to correlate fairly well with the number or percentage of mice that responded. I try not to make too much out of that because of the lack of clear translation from a mouse behavior to a human behavior. So I guarantee you that was not a satisfactory answer, but it's a really hard question.
It's a hard question. I wanted to share that because you never know. But of course, it's a hard question to answer. I think it's just more about safety in the beginning when you're dealing with mice models, and then it is a huge jump to go into humans because if you don't have that safety profile, then it's not even possible to do that. So Anyways, I know you, we've taken an hour of your time, and we really appreciate you spending time with us and just, sharing your information that you have. I wanted to see what you kind of said, what's next. Your second trial, it's already full. You're already in
Oh,
the work.
no. It's just getting underway. So, we haven't even announced formally that the trial is going, but we'll be making an announcement along those lines. We hope, cross our fingers, everything is continues to go well, that we'll be making an announcement like that soon. But I would just encourage people to go to clinicaltrials.gov, and you can see there kind of where the different, well, there's at least contact information that you could get to the people running the trial. And just to give you a sense, this is we'll have about 20 to 25 sites in the US, so all over the US. Most of the big population regions will have a center in there, and then several in Australia and New Zealand as well.
Wonderful. Well, we will also provide, Enrique will add a link onto the group so that people can go in and check and see if their kids fit the age, if they fit the location, if everything, all the boxes are checked. Hopefully we can get some more people in your trials, because I think people are really interested in this. And if there's anything that you need on our side, please feel free to reach out. We have a huge group of people. People are happy to give money where it's needed, and also give their time. So we'd love to collaborate with you guys in any way possible, so we can just help to further your studies, and hopefully to find some answers for so many people all over the world. So, well,
we really appreciate that. And if there is a way to collaborate, let's talk about it, because one of the things that we've learned along the way, there'll be no surprise to the people in the audience, that the autism community is certainly one of the best organized groups of people that we've ever seen, and they're one of the most motivated groups of families. And so I think, if anything, if we can keep up with you, then that would be great. But we'd love to find a way to collaborate.
Absolutely. Well, thank you so much, Stewart, for your time and for your wonderful presentation. And we are very excited to see what you do in the future. So we'll all be watching. Thank you so much again.
Thank you.